Okay. Assalamualaikum warahmatullahi wabarakatuh. Good evening, ladies and gentlemen. Thank you for joining our Researcher and Student Enrichment Program today. Before we start our agenda, let me reinform the webinar protocol to join this event. First, please adjust your name or ID screen using the given format. Second, during the event, please turn off your audio and only turn on the audio when the moderator gives you the chance. Third, please turn on your camera. Fourth, ensure your network has a stable connection. And fifth, adjust your position comfortably and prevent the backlight effect. I will also read out the important information for today. First, please fill in the attendance form on the link bit.ly/attendance_R2SEP 2021. The link will be closed in 15 minutes. Second, please fill in the feedback form that will be shared 15 minutes before the session ends and will be closed after 1 hour. Third, the attendance and feedback is compulsory for these purposes. First, it's mandatory attendance for ITS masters and doctoral students new intake. Second, it's eligible for a certificate of participation, and it is for international participants who have 60% attendance or minimal 5 meetings. And then third, it's for STEM for it, and it is eligible for ITS undergraduate students. Welcome to Researcher and Research Student Enrichment session on today, Thursday, the 20th of May, 2021. I am Nur from ITS Global Engagement, and today I will be your master of ceremony for this session. Please do not forget to fill your attendance at the link bit.ly/attendance R2SEP 2021 that will be sent by our committee via the Zoom chat room, and the link will be closed in 15 minutes from now. Participants who wish to ask questions during the question and answer session, please send your question at the link bit.ly/heart_and-Q&A_R2SEP that will be sent later by our committee via the Zoom chat room. Alternatively, you may raise your hand during the Q&A session to ask your question directly. Before we start our agenda, allow me to deliver our schedule today as follows. For the first agenda is the opening, and then continued by the second agenda, which is the introduction to the moderator and speaker. After that will be the main agenda, the lecture session, and then followed by the Q&A session. And there will be an info session from Professor Amit Agrawal, followed by the Q&A session, and will be continued by the certificate awarding, and finally followed by the last agenda, that is the closing of R2SEP. Before we move on to the next agenda, please allow me to elaborate about today's lecture. Today's lecture theme in Hard Engineering stream of R2SEP is about research management skills and life and opportunities for higher education at IIT Bombay, India, which will be delivered by our speaker, Professor Amit Agrawal from Indian Institute of Technology, Bombay, and Engineer Joendra Bambang Aryateja from ITS will be the one conducting today's session as our moderator. Let me introduce our moderator for today. Our moderator today, Mr. Julian, is a lecturer in Department of Mechanical Engineering at ITS. Next slide, please. Okay. As for the educational background, Mr. Julian finished his bachelor degree in Mechanical Engineering at ITS and his master degree in Institute Technology Bandung in the same major. His research interests include fracture mechanics, numerical methods, and structural integrity. Now that you are informed about our moderator, without further ado, let's proceed to the main agenda. To Mr. Julian as our moderator, the time is yours.
Thank you very much, Nur. Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon or morning or evening, maybe. So we already know we are going to have a R2SEP, which is the speaker is Professor Amit Agrawal. Professor Amit Agrawal is one of the academic and lecturer in Institute Mechanical Engineering IIT Bombay. Wait a minute. I just want to read this one. Joined the Indian Institute of Technology, Bombay in 2004, and currently an institute chair professor in Department of Mechanical Engineering and Dean of International Relations, IIT Bombay. His research interests are especially in microscale flows, development of novel microdevice, and theoretical fluid mechanics and turbulent flows. He has graduated more than 26 PhDs and postdoc and master students, and also he's published more than 200 journals and dozen patents. His primary contribution are in the development of unique blood plasma separation, microdevice, and derivation of equation which he believed are more general than the Navier-Stokes equation. A technique for processing data in turbulent flows is sometimes referred to as Agrawal decomposition. His insight and novel result on transmission of COVID-19 are documented as 7 featured articles in prestigious journal Physics of Fluid. Professor Amit Agrawal has authored a well-received book entitled Microscale Flow and Heat Transfer, Mathematical Modeling and Flow Physics. The work from his lab has appeared on the cover page of prestigious journals such as Journal of Fluid Mechanics and Physics and Fluid. And more of his contribution to the knowledge. And so today he will conduct a presentation. Yes, Professor Amit Agrawal. He will conduct. Hi, Professor. How are you today? I think you still mute.
Yes, good afternoon, and so thanks for having me here.
Yeah, okay. Today we are going to discuss about 2 topics. The first one is about your specialist research, which is microdevice in plasma blood, I think. Sorry for my description is not very good. And then secondly, he will discuss about the opportunity to study further in IIT Bombay. So I think there's no further ado. I give the time for you, Professor.
Okay, so good afternoon, everyone. Thank you, Dr. Jaden, for the kind introduction.
You're welcome.
It's a really wonderful feeling to be here and having this opportunity to talk to all of you. So I would really like to thank Professor Maria for suggesting that I basically attend or come for this symposium that we have every week for the students. And I'm also very grateful to Ms. Nabila and her team who have done a wonderful job in coordinating and doing everything, preparing for this, and Dr. Estria. So thank you all of you for having me. So I will start off with a presentation on my work. So can you see my screen?
Yes, bro.
Okay, so yeah, so I understand that this is a lecture where the young students are sort of exposed to how one should go about doing research. So I will essentially share it as an example of what we have been doing in the last few years. So we have developed some micro devices, and I'm going to talk about in very simple general terms. I understand that there may be people from other background, not just mechanical engineering. So I hope this lecture will be accessible to a wider section of people. So I have essentially two parts right now. So I will be talking about a little bit in depth about how we went about making these devices, micro devices. And then I will also give you an overview of some of the other things that we do. And then in the second part, as Mr. Julind has said, we will be talking about opportunities and life about studies at IIT Bombay. So please allow me to start off with this first part, which is development of some innovative point of care micro devices that have been developed in our group. So let me start off by acknowledging the students who have been actually the workforce, and they are actually the ones who have actually done all the hard work.
Sorry, I cannot hear you.
Okay.
Okay.
Yeah, so I think I got muted somehow. So there are a few micro devices that I will be talking about. So these are the students who have actually made these micro devices during the course of their PhD work or the master's work, and sometimes even as part of the postdoctoral work. So we have received funding from the Department of Science and Technology in New Delhi and Vadwani Research Center for Bioengineering at IIT Bombay. And I'm grateful to these funding agencies for the money that we have received. So let me start off by talking about the broader objective. So as we know that there are a few devices in the market which are very simple to use and which can give us an idea about the glucose level that the person is having in the blood, or whether the woman is pregnant or not. So there are several kits like these which are there in the market. And as engineers, our objective is to be able to make these devices which are simple to use, they are cheap and quick, they can get quick results, and they involve a small amount of sample and reagents for their analysis. So if you were to look inside one of these devices, like what you saw in the previous slide, you would see that there are going to be several modules which are going to be there. For example, this is a device that detects that detects dopamine from whole blood. This is an example which is coming from the literature. You will see that there are two main modules. This is one module and the other one, this is the other module. So in one of the modules, you would see that blood is taken. This is, you are trying to detect dopamine from blood. So blood is coming in and you essentially separate out the plasma, which is where the dopamine is supposed to be. And then in the second part, you take it to a sensing module, where some optical sensing, as you can see from the light, which is going to be passed, it detects the amount of dopamine level that is there in blood. So I like this example because these have actually used our blood plasma separator, which is this device here that you see, for doing this, for making this device. Okay, and I'm going to talk about how we went about actually making this blood plasma separator. So you can see that there are several things that you need. You are taking in blood, you want to measure dopamine, but there are several processing steps that you may need to do. So the blood may be taken in, and the plasma, which is where the dopamine is, has to be first separated out, and the cells and other interfering things that can interfere with the signal have to be removed. So there is a separation module and there is a sensing module. So we'll be talking about only the separation module, which is something which involves flow of blood, and which is where our specialization of understanding how they flow, what are the forces which arise when there is a flowing fluid, which is what is of interest, and that's what we'll be using here. So I will be talking about in the first part about how we went about designing this blood plasma separation device. So first, why do we need plasma from blood? We have already seen an example. And if you go to a doctor, if the patient is sick, one of the first things that is advised by the doctor is to go for a blood test, because blood test is such a good indicator of how the human body is functioning or where the disease could lie. So, but if you want to do the blood test, the blood is drawn, a few ml of blood is drawn from the patient's body, and then it is centrifuged. The plasma is separated out. The cells are then thrown off, and then the plasma is taken for further testing. But now, if you want to think about the making a small device like what you have, you'll have to do all this in the case of in a small micro device and a small platform. Okay, so just to remind you about blood and its constitution, or the constituents which are there in blood. So the blood has a number of cells which are called as red blood cells, RBCs, white blood cells or WBCs, and platelets. So the RBCs constitute around 45% by volume. So if you take the blood and let it sediment, you would find that bottom 45% or so by volume will be occupied by these red blood cells. And these red blood cells are very large number. There are around 5 million of these cells per microliter of blood. And then they have around 8 microns as the size. And around WBCs and platelets are around 1% by volume. They will be essentially occupying this white region here. And then they are much fewer in number, as you can see. And then we have the remaining part, which is the liquid part, which is the carrier fluid, which is a carrier which is carrying all these cells when the flow is happening, when the blood is circulating in our body. That's the plasma. And this is where all the information about the body lies. Most of the information lies in this plasma, the liquid part. So we need to separate out the plasma, and the way it is done, as I had just said, that we take a few ml of blood from the patient, centrifuge it, and then these components are separated out. But here the issue is that we saw that we have a very small device. We saw that that's what is of interest. That's what we would like to make. And we cannot integrate such a large centrifuge with a small device. Okay, so we'll have to do the separation of plasma from blood at the chip level itself. And this is where a lot of different companies are trying to work on. And if you can separate out this blood, then you can take it to the second module, the sensing module. And then you can detect anything like what we saw in dopamine, or it could be glucose level or anything else that is of interest to you. And that can be easily done. So let us just look at some ideas. So if our problem statement is that we want to separate out the plasma from blood, can we just not use some sort of filtration technique like what has been shown here? So one can use filter, but the issue is that these filters tends to get clogged in very little time, and therefore the efficacy or the amount of plasma that we will get out will stop. The plasma will stop coming out after some time because this will clog. And the way to see that is, we said that there are 5 million of these RBCs per microliter of blood. So you can see that if you are working with a few ml of blood, then you are talking about a few billion of these cells, and they are very quickly, they will form a layer and they will stop the flow of plasma from the blood side to the other side. So that's why people do not go with filter-based technique. People have tried to use other type of techniques, like what you see here, where blood is taken in, and you have some barriers to stop the flow of cells. And then the idea is that only the liquid part, the plasma, will ooze out, and it will get collected here, and the cells will go on the other side. But here, again, it's an idea of using mechanical barriers. So the issues that we were talking about with respect to clogging, with respect to the analyte of interest getting absorbed on the filter surface, all those issues stay here. They remain. So this is not a very good idea. So people have tried to use alternative techniques. There are a lot of people who have worked on this problem, and a lot of different designs are available in the literature. So we will not really go through all of them. I'm just trying to give you a cross section of what exists, so that you get a sense of how one could approach this problem and what path to take and what path to avoid while learning from the experience of the previous researchers. So here you can see that this is another device that was proposed by one of the groups, where you have blood coming in and you have all these interesting features, very complex features that have been built in. And then you separate out, you put in blood in this line, blood flows in this line, and this is where the plasma is supposed to come out. After passing through all these passages, and the idea is that we are going to rely on hydrodynamic forces for the separation. So, but the issue is that if you want to use this device, this is to avoid, I mean, you put in one patient's blood, you do get the plasma out, but then you cannot put a second patient's blood because there could be cross-contamination that can happen from the first patient to the second patient. So you have to avoid this, you have to throw off. So you can think of that if you have made such complex shapes and passages, it becomes very difficult. The cost of making these becomes large. And therefore, it is not very economically viable to keep on throwing devices like this every now and then. So we have to sort of, so we can see that this is the background in which we started doing our work. We realized that there are small dimensions which are involved. You can see that 20 microns passages are involved. Cell size is 8 microns, so you can have a few cells coming together and that will clog the device. You can see that these are the issues that that you see with the existing devices. You can also ask the question, can these, instead of having such a complex shape like what you saw in the previous, can you not have simpler designs which are also effective and maybe better than, give you better performance than what has been demonstrated in the literature? So with that as the background, we started doing our work. And then before we start describing about how we got a solution, we have to understand what are the physical principles which are involved, which can be utilized for this purpose. So here I will show you a few principles which we have used. The first one, which we call as a Fahraeus effect, which is when if you have flow of blood happening in a small passage, what we would find is that there is a cell-free region that is formed, and this is cell-free region. So what you would find is that the cells, the RBCs, will tend to flow towards the center of the channel, and there is a small region which is known as a cell-free region, which is left on the walls of the channel. So you can think of this as the shear-induced migration of particles of these cells, which is happening. So the region of minimum shear is at the center of the channel. So the cells tends to move away from the walls and move towards the center. So this is one effect which kicks in at small scales, especially 200 microns and smaller. So we can use this effect. This is one of the effects that we've utilized in our separation principle. And then you can see that near the wall, and you can see from the lighter shade of color which is available, you will be able to draw out this plasma, which is relatively cell-free. Now we can also have another effect, this so-called bifurcation law, where if you have the cells coming in at a bifurcation, they have an option of going towards the left branch or the right branch. And it is known that the cells will prefer the branch which has the higher flow rate, provided these conditions are met. So this is another important, interesting phenomena that people were aware of, which could be utilized here to make our device like what we are interested in, like trying to separate out plasma from blood. So putting these two effects together, we could actually make a very simple device, which we call it as a T-channel microchannel, where we can get some plasma coming out. But this has been demonstrated with diluted blood. So you have to take blood, dilute it 20 times. And if you do that, you can go with a very simple design like what has been shown here, and you can get good quality plasma coming out. But this is not what is of our interest. We want to actually not just work with diluted blood, but blood which has been directly drawn from the patient without the need for diluting it. So then we have to do something else. And what we realize is that if you pinch the passage, that means the cross-section of the passage certainly reduces. So what would happen is your velocity will increase substantially in this pinched region. And then once you release the pinch, the cells will continue to move with a much higher velocity, which they have acquired in this region, in the pinched region. And then the liquid will have its own trajectory, and these cells would have its own trajectory because there's a difference in the velocities that they would have, and because of which there would be separation that they would have. And this is a region of cell-free region that you can see, and you can utilize this corner and get some plasma coming out from here. So, and we also realize that we can bring in geometrical effect like what you see here. Just bend the channel, bring in the centrifugal forces, so the cells will tend to move out towards the outer side. And then in the inner side, from the inner side, you should be able to draw out this plasma. So we realize that these are all, you can see that these are all hydrodynamic forces. These are forces which are happening because when the flow is happening, and it's just that these forces are acting differently on the cells as well as the plasma, and therefore that is what is leading to this their movement along different paths or in different ways, and then we can utilize these effects for our purpose. So we essentially, after realizing all these possibilities, the idea was to put all these different effects together in the device. And what we see here is there is blood which is coming in. Now this is undiluted blood. You can bring in from the lower side, take it through this passage. We have seen that bending brings in centrifugal forces. You can see that there's a bifurcation. You can see that there is also an expansion. So we have seen that these are the features that can help us. So we have put all these features together in an optimal manner. And this did take us quite several years actually to figure out the optimal sort of configuration or optimal way to do these things. Although we understood the principles much earlier, combining them together took us quite some time. But ultimately, we are happy with the way that you can get things. So you can see that we have whole blood, undiluted blood coming in from this inlet, and you have very high quality plasma. You can see almost no cells which are flowing in this passage. Everything is flowing towards the left here. And so this is, and these are all 100 microns plus sort of dimensions. So these are relatively large dimensions that we are working with. So there are no issues of clogging, etc. with these devices. So this is, and one can do a detailed characterization of what you're getting, and you can show like what has been done here, that if you take blood and you just pass it through the cytometer, you would find that this is where the RBCs and WBCs will come in. This is the window. The platelets will be coming in this window. And what you see here is with our device, once you pass through them, this window is almost empty. I mean, a lot of cells have been removed. And this is, gives you, I mean, very good quality plasma is what you get from the device. So this is some comparison just to sort of show that how we compare, our device compares with what is available in the literature. And what you will see is that we do better on all the counts that one could compare against. So I will skip this and say that there are several unique features of our device, which can give us almost 100% separation efficiency, not just with the whole blood, but even if you have something which is have still higher number of cells because of certain diseases, our device can still work. And you can process a large amount of blood with this. You can process 3 to 5 ml of blood in 10 minutes, which is actually very good. There are no issues of clogging and so on. So this is a very robust device that we have, and you can see that this device is a point-sized device. And so you can essentially carry it in the pocket and all. So it's a very simple device. Took us several years to come out with, but then once we have this understanding, we can actually use it for other cases as well. So this was the first device that I wanted to describe, and I just wanted to describe how we went about this, making this device, explaining the physical principles, putting it together, and so on. So now once we have this device, a blood plasma separating device, we can be a little bit more ambitious and say that we don't want just plasma, we want platelet-rich plasma. So platelets are something which are very useful. When the patient has dengue, for example, it's only the platelets that needs to be administered to the patient. So the donor need not give the entire blood. You can draw blood from the donor, remove the platelets, give it to the patient, and put the remaining blood back into the donor's body. So that sort of a thing one would like to do. So what we would like to do is get platelet-rich plasma And of course, in certain cases, we'd also like to have platelet poor plasma from blood coming out. So what we could do as an extension of this work was to draw these or to make this platelet-rich plasma suppression device. So I've already described the basic principles on which this device can be built. So, so we have, you can see that this is the type of device that we came up with. We have a flow which is coming in from one side, and then there's a trifurcation that is happening. The blood flows, most of the blood flows here, and at the sides you would see that you have plasma and a lot of platelets coming out from the side. So again, designing these passages is where, which takes some time, and this is where the student has to do a lot of iterations to understand what design works well. And we realize that the presence of RBCs is very important for suppression of platelets. So those are some interesting insights that we got. So I will not really give you all the details, but I will just give you the final sort of results and say that if our target was to enrich the platelets by 3 to 8 times, this is what is, so if you can demonstrate that you have 3 to 8 times the physiological level of platelets in the plasma, that is called as a platelet-rich plasma. So here we can show with our device, and as I said that there were several devices that we made, and we could show that we can actually get even 14, 15 times enrichment, which is better than what we need. So we can definitely be, this plasma that we're getting is actually a platelet-rich plasma. So it's coming with a lot of platelets, and at least 15 times enrichment has happened with respect to the number of platelets that we're putting in. Now, we also did biological characterization of these platelets to show that these platelets can be used for the purpose of transfusion. So we, here you could see that there are 3 curves. One is that if you take the inlet sample, this is the green curve that you see. Now, when you centrifuge it, is the red curve that you see. And the blue curve is the one that is coming out from our device. And what has been shown here is what you want is that you should have the curve the same as the green curve. And you can see that once you centrifuge it, there is a lot of deviation of the red curve with respect to the green curve. And this is a known problem with centrifugation, that it activates 50% of the platelets. So that means 50%, only 50% of platelets are useful after centrifugation. Whereas what we see from our device is that the blue curve is almost overlapping with the green curve, and less than 6% activation of cells happen in our case. So what we are demonstrating through this curve here, through this result here, is that this micro device that we have built is actually gives you better performance than even a centrifuge, which is actually regarded as a gold standard in some sense, or it's the only way to obtain platelets from the patient. So this micro device can actually do a better job than the platelets, than the centrifugation itself. So, so these were the 2 problems, the first 2 sort of problems that one can attack. And then as a further extension and further, so once you sort of understand how to make these devices, you can do other things with that. So let me describe a third problem and sort of give you how we again went about finding a solution for that. So I will talk about hydrodynamic focusing device. So this device is, if you take red blood cells, if you take blood cells or blood, you would like to know whether there are other cells, which may be like cancerous cells or could be other foreign cells, which are indication of some disease in the patient's body. So if you want to know that, you have to take the blood and pass it through some sensor. And then the sensor is going to continuously monitor the cells which are passing through it and recognize them as either the cells which are known, red blood cells, white blood cells, platelets, etc. Or if there is no match with this database, which could possibly indicate that there is a foreign cell which is there in blood. So if that is a situation, then it should flag and say that there is something wrong with this patient, and maybe it needs to be due to further screening. So now so that is the problem statement, that you need to be able to continuously scan the blood, see if there are any foreign cells, and if there are foreign cells, can you flag it? So Now, one of the aspect again is to make the scanner. The other aspect is to be able to do this, to be able to facilitate this, to be able to make a micro device, which can do, which could facilitate this scanning of the cells. So that means that, what it means that you can see that if I take a scanner here and pass light or whatever other detection through this cross section, I hope you can see my cursor. So you can see that it is going to pass through all these cells, and if the cells is hiding somewhere here, a foreign cell, I will not be able to detect that. So the idea is, maybe if I use a focusing device and put a scanner on the other side, then then the number of cells through which the sensing is happening is much lesser, and therefore the ability to detect a foreign cell increases substantially. So you can see that that's the basic problem statement that we are making, that you should be able to take the sample and focus it. Now, the focusing can happen in a very simple thing like here, but this is not the only thing, because here the cross section, along the cross section, it is not getting focused. So it's only getting focused from the two sides, not from the other two sides. So if you can focus it from the other side also, then that's essentially what you would like to have. So if you can focus it from all sides and have a very small spot coming out, like what I'm showing from my hand here, that's where, and you can just put your probe there and see everything which is flowing. So that's that's what we would like to have. And if you can make a device like that, we'll call that as a 3-dimensional focusing device. So, and the requirement is that you would like to have, here you can see that I need 2 pumps. One pump is to pump this fluid. The other pump I need to have to pump this secondary or the surrounding fluid. So I need 2 pumps, number of accessories increases, and I don't want to do that. So can I do, so I would like to ultimately be able to design things which have minimum number of accessories, a number of external components, devices supporting instrumentation which is required is minimum, and so on. So you can see that again, we have some very interesting requirements, and it's a very interesting engineering problem that one can try to solve. It involves some amount of research, because we have not really, there is no ready solution that is available. So we'll have to now think about how to go about designing a solution for this case. So what we could come up with is a design like what you see here. So it's a very simple design. You can see that we saw that there are always going to be 2 fluid, one which is sample and the other one which is the secondary or the surrounding fluid. So you have 2 inlets. We will have minimum of 2 inlets. You will need minimum of 2 pumps. So A and B are, A is where the sample is coming in, let us say it is blood, and the B is where the surrounding fluid is coming, which is the sheet fluid. And then they are passed through this, after passing through this common section, this is where, this is how they will sit. The flow rate of B is much larger than the flow rate of A, so it will occupy a much larger area, which is what you see here. And this is where the sample B is sitting. Now, what we do is simply pass it through 2 curves. This is very interestingly designed curves which have been put in. So if you pass it through this curve, you see that this vertical line becomes a horizontal line, and it happens because there are some Dean's vortices which comes in when you take a flow and pass it through a curved passage. So that's the effect that we bring in here. And then you bring in the pass it through a second curve. So this, you can see that this vertical line has already become a horizontal line. And then once you pass it through the second curve, you bring in the centrifugal force and you pass it, and you can see that this elongated thing becomes a tighter spot, and that's what you want. So you can see now the surrounding fluid is actually surrounded this cells from all sides. Okay, so this is what one can see. And this is how, this is a slide which essentially illustrates the basic principle of operation of the device. And so this is the device being made, inlet for the secondary fluid at this primary fluid. And this is the outlet. And you can see that this is the region where I can actually put my sensor. So I can put my sensor in this part and start focusing, start probing my cells, which are going to pass through to the spot that I have shown earlier. So this is, if you were to do experiments on this device, this is what you would see. And so you can see that you would get these spots that I'm talking about. This is where the cells will be flowing. And the dark region is where the surrounding fluid will be there. And you can then probe these cells quite well. So you will have only 3 or 4 cells which are moving in this spot. Of course, you can try to make this spot still smaller. That would be of 1 cell width, that would be ideal. But what we could demonstrate is around 3 to 4 times the cell width. And of course, this is some comparison simulations and experiments that I would skip. And then if you were to take this device, pass the blood through this, you can see that you can get focusing, and you can see it from 2 different views, the top view and the side view. And you can see these cells, very thin cells, I mean, or the thing passing it through these lines, which is where the cells are passing. And of course, the 2 views confirm that this is actually getting focused in the 2 directions as you would like to have. So this was another device that I wanted to talk about. And so again, this is a simple device to fabricate, which has been, as you can see, that there were only requirement of 2 pumps. And so you can see that these are the devices which have been found very useful for certain companies and microfluidics, the people who are doing microfluidics. So essentially we gave the solution to 2 different microfluidics companies. In one of the companies, they actually put all these devices together, and they came up with several unique solutions of their own. So these are the type of things that one can do if we were to look at this problem from the fluid mechanics point of view. So you can see these are all fluid mechanics based solutions, and what you are learning from the basic fundamentals of fluid flow can be used here in these cases to make some innovative devices which can again solve some interesting problems. Now, as another example, I will talk about a heat transfer related application. And so the problem statement is that when you work with these samples, these biological samples, the body temperature is 37 degrees Celsius. The room temperature is 25 degrees Celsius, or or it can vary. I mean, lab temperature is typically maintained at 25 degrees Celsius. So you don't want the sample to be maintained at, so sometimes you want the sample to be maintained at the body temperature. So for that, if you have to heat up the sample so that you are trying to maintain it at 37 degrees Celsius. But if you heat up the sample, then there is a chance that you overheat and you damage the sample itself. So how do I solve this problem? So how do I make sure that I heat it enough so that I maintain the sample at 1 temperature without the possibility of overheating and destroying the sample? So the idea is very simple, and this any person knowing heat transfer knows that we can try to maintain the wall at a constant temperature. So if we maintain the wall at a temperature that is required, then we know that there is no possibility that the sample will get destroyed. So that was the idea. So the idea is that can we make a device which can maintain its temperature, which can maintain the wall at a given temperature. Now, if you were to think about this problem, this is not a simple problem statement, because this is done in the lab and other places using a phase change material. For example, if we take water, boil it, we know that water will boil at 100 degrees Celsius at 1 bar pressure. So that's the, and that's when the boiling is happening, the temperature is maintained constant. So the phase change is what gives you a constant temperature, but having a phase change solution for maintaining something which is a constant temperature is not always feasible. So for example, in this case, we want something to maintain at, the wall should be maintained at 37 degrees Celsius. So are we going to find, are we necessarily going to find a phase change material, a material which is going to change its phase at 37 degrees Celsius? So you can see that that is not going to be very easy to find a material like that. And can you now maintain any temperature, not just 37 degrees? If I say now tomorrow my requirement has changed, I want to maintain it at 42 degrees Celsius. Now what will you do? So you can see that I cannot keep on finding different type of phase change materials, and I will have to rather look at the solution from a different point of view. So this was the objective of what we started doing. And we started thinking that, is there a way to combine certain effects again. We sort of came up with the idea of conduction, combining conduction and convection, like what has been shown here. So let me explain this slide and explain the idea behind what we are doing here. So let us say that, okay, so this is all based on the observation that most of these micro devices have a huge thick wall. Okay, so the thing is that if you have a thick wall, like what has been shown here, this is my wall, and then this is my passage for the fluid to flow. So because of the thick wall, because of the conduction which is going to happen in the wall, there could be, so there is conduction in the wall, which is also very important, and there is convection which is happening on the inner surface. So there are these two effects, and the idea is, can we combine these, or can we utilize this in a certain definite manner and be able to maintain things at a constant temperature. So let me explain this through this example here, through this figure here. So what we're doing here in this case is supplying heat at a constant heat flux at the bottom outer wall. And the fluid is moving in from the left to right. So from the left to right, as it comes in, the cold fluid comes in. So this end is cold. And by the time the fluid leaves, it has picked up heat from the wall and it has gotten heated up. So you can see that this side of the wall is going to be heated up. So if you look at the conduction would happen in the reverse side, because this, from the point of view of the wall, this is heated up and this is the cooler side. So the conduction of heat in the wall would be in the opposite direction to the flow of the fluid which is happening on top. So once we realize this, and we realize this conduction is very important, you can actually play around with the relative strengths of conduction and convection and try to see that make this linear increase in temperature with position to a more flatter T versus X sort of a curve. Now to change these effects, one can change the wall thickness, one can change the flow passage, the geometry of the flow passage, so that the convection, the strength of convection changes. So that those were the ideas with which we started working with. And we realized that to reduce the convection, maybe we can have a diverging passage, like a diverging channel. So we essentially made these diverging channels. We also did numerical simulations, detailed numerical simulations to study this problem. So what you could see here is that this is the passage, and these are the points are where the thermocouples are going to be located to measure the wall temperature. These are special thermocouples which have very small bead size, so they can actually give us the local temperature, wall temperature. So you could see that in this diverging sort of a passage, if you look at T versus X, you can see that T versus X is actually very constant over a very large percentage of the channel length. Okay, and it becomes constant because of the effects that I talked about in the previous slide. Similarly, if you look at the temperature variation in that lateral direction, you can see again they are very constant. And of course, this is the same thing which has been shown using infrared imaging. We have an infrared IR camera which was used to image the surface, and you can see as you keep going towards optimal conditions. So initially you have a lot of temperature variation, and you can see all the colors from all the way from blue to red. But once you come to the optimal conditions, you can see it's mostly green. It's only a single color, which means that the wall has now attained the constant wall temperature condition. That is what we were talking about. So one can, of course, ask this question that is just a fluke that we got a good balance between conduction and convection and temperature became very constant, or this is something which is applicable to a larger parameter range. So to demonstrate that, we did a lot of simulations, three-dimensional simulations with property variation. And because there are a large number of parameters in this problem, we have the divergence, you have the flow rates, you have heat fluxes, you have material conductivities, thermal conductivities, the fluid properties. So we have a lot of parameters. We have to optimize all them. So we essentially followed, we first validated our simulations by comparing against experiments. That's what you see there, the curves and the dots. And then once we have good simulations with the property variation, etc., being put in, we started predicting how the effect would be if we change the angle, if we change the length, the depth, and other parameters. Conductivity ratio. So we could actually do a very detailed simulation with all these parameters. And then based on that, we could sort of understand what affects more, which parameters have a stronger effect, and so on. And we could also show through this exercise that is actually the solution that we're talking about is actually applicable to a large range. It's not just one point where the two curves intersect, but this solution is actually applicable to a reasonably large parameter range. So this is actually something which can be utilized in practice. And then one can ask the question, what is the temperature value that it will take? So we came up with a model to predict that. And you can see that the model gives you, if you were to compare the model predictions versus the experimental predictions within 1 degree, you can see that there is a match between the two of them. So, I mean, one can use this idea to make a PCR. Now, with COVID testing, PCR has become like a household term now. And so PCR essentially is a thermal cycler, which takes this RNA and DNA molecules, and they just DNA strands and essentially breaks it down and then recombines it. So there are 3 different temperature stations. So as an engineer, the objective is to be able to design the temperatures and the residence time for these samples. And what we've been trying to do is to have these 3 stations and maintaining things at the temperatures which are required. So what we could show through these simulations is that in principle, at least we can have these PCR. So instead of having this PCR, a sophisticated sort of a PCR, we can actually have a much more, this micro device based PCR if you want. And you can have all the advantages of these micro devices that we have talked about. So, of course, there are some interesting questions that can be asked from the fundamental point of view. I think I will skip these 2-3 slides. I think I have still a good amount of material to cover. So I will sort of come to this. So I want to come to the sort of end of this part where I just try to give you a little bit more in-depth information about our devices and how we went about designing them. And as a quick recap of what we have said, we have talked about the blood plasma separation micro device, where we could demonstrate that we don't really need a centrifuge. We can actually do everything that a centrifuge does at a micro device. We could also show that it can be extended to extract platelet-rich plasma if you want. We also talked about the 3-dimensional hydrodynamic focusing device, and we said that it's a much simpler device which requiring much fewer accessories, and based on a very simple design, which is, from the concepts you can understand why it works. And we also looked at a constant wall temperature device from a very thermal point of view. And we said that we can actually take this constant wall temperature device, put it together in the form of a PCR. Of course, requires some more experiments to show, but through simulations, we've been able to demonstrate. Of course, experimentally, we have demonstrated that a single device, a single channel does give you a very constant wall temperature. But if you can actually put that together in a PCR, that is something which is still a work in progress. And we would like to sort of demonstrate that as well. And then you can take a PCR, micro PCR at this point. Now, what we would like to do is very quickly give you an overview of the other things that we do. And so I will just switch. So I hope the second presentation is clear. So the slides, I'm just giving you a very quick overview of what are the other activities that have happened. And the idea is that we will also sort of see that there is a lot of collaborative work that has been happened. So you can see that the work of other colleagues also, at least they have indirectly contributed, they have contributed by being a co-advisor of some of these works. So this will be a very few, slides. And of course, you can see that each of these slides is actually one PhD work. So it's actually a lot of information which is behind it, which I'm all skipping together. So I'm just going to give you a sense of what are the other activities that happens. So because we have been talking about microchannels, so I will start off with microchannels and go ahead from there. So this is a work which was done almost a decade back, where we were actually looking at boiling in microchannel. So boiling in microchannel was a very interesting problem to look at, because here, as you can see, this is a 100 micron passage. You can see these bubbles which are going to form, and you can actually image these bubbles as they form, grow, and get enlarged, and then they fill up this entire passage. So you can actually visualize all that. We have enough videos of that happening. And then you can see that these bubbles that form, they tend to coalesce. Several of these bubbles tend to coalesce, and then they will form a bigger bubble or a slug. And we could actually visualize, and then at higher heat fluxes, it will become an annular flow. So one can have different flow regimes that one knows in two-phase flow. One can actually look at how these different regimes comes about. And we were essentially trying to do it from the point of view of electronic cooling. We wanted to see whether we can use this. We have electronic chips on one side. We can flow water, let the water pick up heat, which is being dissipated by the chip. And then the chip cools down and the water can even be allowed to boil. And then this water can come back and pick up the heat again, and so on. So this is the types of the cooling solution is what we are looking at here. And so from that point of view, we're looking at how much will be the pressure drop, because this is like the penalty that you'll have to pay. You'll have to do the work. Your pump has to do the work to sustain the flow. So this is a penalty. So you have to keep an eye on the penalty and look at the performance that you would get. So this was a detailed characterization of the flow regimes that you would have, the flow map we made. Based on the information that you get here, we have to characterize how much heat transfer coefficients we would get, what will be the pressure drops, and so on. So I mean, so this is the type of work, one of the works that we did. In fact, the student who did this work now is a professor in IIT Hyderabad. Now moving ahead, because we're talking about electronic cooling, so I will now talk about a very different sort of a solution that we have been looking at. Now this is a work from another PhD student, Mangesh Chaudhary. So here you can see that that turbulent flows. So till now we have been talking about micro scale flows, which are all laminar flows, whereas large scale flows like flow in the atmosphere, flow in the ocean, these are all turbulent flows. Flow becomes turbulent, and turbulent flows have some interesting properties. For example, they can be used for removing much larger amount of heat. They have typically much higher heat transfer coefficients. So one can use these properties, and make a synthetic jet, which is a small diaphragm, which is just oscillating, and you have flow coming in and moving out of this cavity. And once you move out, it is like breathing. You just see that there is a jet which is coming out when you push this membrane back up. And once you, so essentially when you draw move the diaphragm from the upper side to the lower side, there is a low pressure region which is created inside the cavity. So the fluid will try to fill in from all sides. And then once you push the diaphragm back up, then the fluid will actually go out in the form of a jet. So this is a synthetic jet, and we were among the first people in the world to be trying to look at the heat transfer aspects of how much heat removal you can have. And so this is the type of flow that you would create. And we took this flow, impinged it on a surface, and we're seeing how much heat transfer coefficient that you will get there. And one can try to again put it on your motherboard if you want, and see what is the type of cooling that you would get in presence of a heat sink or without a heat sink, and so on. So this is the type of sort of work. Again, this is a relatively old work, although several students have worked in my group on this problem, looking at the heat transfer as well as the flow characteristics of these flows. Now, when we talk about turbulent flows, we typically use PIV as a technique. So we have a PIV which is there in our lab. And I'm just showing you a different example. This is an unpublished work, but this is an interesting example, and students here should probably like that. So one of the students actually just used this, put a fish in a fish tank and put a PIV and just started to look at what will be the flow around this swimming fish. So this is a live fish which is swimming in a tank. And the idea was to see what sort of a flow pattern is formed around this fish. So you can see that this is a flow pattern which comes out. You can draw the vorticity, for example, around this fish. You can see what is the type of disturbance that is being created by the fish. And you can see that there is almost no disturbance that the fish creates. And of course, this is important because it does not want to be, a smaller fish does not want to be detected by larger fish. If it leaves a lot of signal, a vague signature, then of course the bigger fish will catch it and it will fall prey to that. So you can see that there's very little disturbance that is created. You can see that as the fish turns, what is the type of flow again that you would get. So I mean, the students essentially spend the entire night because the fish is just moving around merrily in the tank, and you have to just wait patiently for it to come in the observation window. So whenever it comes in the observation window, you get some data, and otherwise you have to just keep on waiting and waiting for it. But of course, you get some very interesting insights about how the swimming happens. So this is just, I'm trying to sort of show that using the PIV technique, you can do study the turbulent flows, and you can also do some fun problems like what has been done here. Now moving on, just moving on, a few more examples, maybe in the 4 or 5 minutes I will take. So I will basically talk about these. If you put a body in the flow, you would have heard about the vortex Kalman shading. Vortices form behind the body and they are shed. So our question was, if you have multiple bodies, how will the vortex form? How will the interaction happen between the wake which is created behind each of these bodies? So we essentially got very interested in this problem, and we started studying this problem. And this problem would have implication in why the fishes swim in a certain formation, or why the birds fly in a certain formation. So we know that one bird or fish is behind the wake, in the wake of the other one, and it is somehow helping that being present in the wake, somehow helping the bird in the wake save its energy, its flying energy. So how does it happen? So it has something to do with the way in the interaction, in which the interaction of these wakes happen. And so this is the type of problem that we were studying. Putting the bodies, these are various cylinders which are there. These are all bluff bodies, each of them creating a vortex shading and then also having interacting waves. So this is a very interesting nonlinear dynamics problem if you want to look at, which one gets. And yeah, so we have several sort of work coming out. And then as an application of this, we did it with one of the colleagues here, Professor S.P. Prabhu. So we were doing the numerical simulation of this problem with a colleague, Professor Atul Sharma. And then with the Professor Prabhu, we were trying to see if we could use this as for a vortex flow meter. So a vortex flow meter is nothing but is used to meter how much flow has passed, how much flow, what is the flow rate in this pipe. If you want to do that, what you do is put a bluff body like what we have shown here, in the like what is shown here, put it, and then you put a sensor. So as the vortex shedding happens, you have a region of low pressure. So that you put a pressure sensor at the optimal location. So this will sense the rise and fall of pressure as the vortices pass through this sensor. And then you essentially look at, from the pressure signal, you find out what is the frequency of vortex shedding. And then once you get the vortex shedding, you can find out what is the Strohal number, like what is given here. And I mean, essentially what you do here is essentially get, because the Strohal number is very constant over a large range, you know what is the value here. You know what is the body dimension, you know what you have measured the frequency, and therefore it gives you the average velocity. So this is essentially what we use here. And we essentially made a number of contributions here in terms of how to increase the range of these vortex flow meters, and how do we get better quality signals, etc. So we had several contributions in this problem, original contributions, which have been patented, for example. And we also have been looking at low pressure gases. So these are very interesting flows which exhibit compressibility effects and slipping at the walls. So typically we think of the no slip condition, no slip, wall slip boundary condition applying, but here that condition gets violated. And you can see that there are some very interesting flow behavior that you get in these cases. For example, if you take a tube, two tubes of two different cross section, you will see that you expect to have a corner eddy forming, but here you see that that is not forming. You will see that there is a sudden change in the behavior of the pressure versus X. These are all experimental measurements that have been made in a tube at low pressure with low pressure gas flowing inside them. So you can see that there is a very distinct behavior of pressure upstream and downstream of this junction. So, so there are some very interesting sort of behavior that one can see. and one can study. And then one can actually try to model these problems, and then you realize that the Navier-Stokes equations itself are not adequate. And then one has to essentially go to some higher order transport equations like the Burnett equations and the Grad equations. So we have some original contributions again in this problem. And if you are interested in understanding about these equations in more depth, we have actually a book which has come out recently. So this is a book that one can refer to. It's titled Microscale Flow and Heat Transfer, but there is a lot more discussion on Burnett equations and such things. So this is pretty much coming to the end. Of course, we cannot in today's time end without talking about COVID. So this is some work that we have done about transmission of COVID. So we were asking several questions about how coronavirus survives on surfaces for days. So this is one of the early reports, which I'm sure a lot of people would have seen, and people, because of which there is, tendency to clean the surfaces, etc. So the question that we've been asking is, how come coronavirus survives for days? And we also have tried to quantify the benefit of using a mask and the probability of infection if you have a super spreader. So some of these results you see here. For example, if you look at, so we essentially what we did is, because we have been working with jet theory, so we can see that if there is a puff cloud which is coming out, we can estimate what is the volume that we would have that is breathing out. And it can be expressed as a very simple formula that has been given here. This is a drive thing, comes from the theory of turbulence. This, although it has been reduced to a very simple form, it becomes as d cube upon 150, where d is the distance which has been traveled by the puff cloud. So you can relate it in that terms as the volume of the puff cloud. And you can see that if you have a mask, for example, the volume of this puff cloud reduces by a factor of at least 7 for surgical mask and 21 by a factor for N95 mask. So, I mean, proportionately, the amount of infection that you are transmitting reduces. And so this is sort of one of the quantification of the benefit of using a mask. Similarly, if you were to look at the probability of infection about from a normal person versus if it was a super spreader, we can see that the probability of infection increases by around 185% for a super spreader as compared to a normal person. So again, this is from some certain calculations about probability that one can see. There are some well-known models which we have applied to this problem with the experimental data as the input, and one can calculate this probability and find out what is the increase amount of infection probability, chance of infection that we have. So this is, I mean, of course, in COVID times, if you work on COVID, you get far more visibility. This is what happened to us. So our work got covered in starting from New York Times to a radio show in Korea, live radio show in Korea, and so on. So we had several sort of several press coverage and such things happening and tweets happening. And of course, this is a message for the younger generation here. If you sort of work on contemplative problems, you get far more visibility for your work. So I think this is all that I have. As I have sort of indicated that I may take slightly longer, but I think I have sort of completed an hour or so. So I will sort of stop here in the first part, and of course in the second part, in the post question answer session, we will talk about IIT Bombay and the opportunities of studying in IIT Bombay. So I would like to sort of acknowledge all the students in my group who have actually been doing all the work that I presented here. So thank you, and I will.
Thank you, Professor. Very good subject. I think it's very interesting and novel. Yes, we have a lot of questions here. Maybe the first question, maybe I will pick one by one. Yeah, the first one is from Astria Nur Irfansyah. She asked about, you talk on plasma aspiration from blood means that the research on your group is multidisciplinary. Can you share how your research group manages this multidisciplinary topic effectively among the members of the group? I think it's about the management of the research.
Yes, actually a wonderful question. And so let me tell you that we do draw expertise from several people. So let me just say that although we are from mechanical engineering, most of the examiners of the student come from bio school, which is also housed in our institute. We do all this fabrication and characterization using microelectronics facilities. So we have a center for excellence in nanoelectronics. So we essentially go there and make our devices. We then go to a central facility, which we have which we have SCM and other tools which are there. The flow cytometer, for example, is housed there. So the students go there, and honestly, I mean, the students have to learn all these various tools and techniques, but there is enough support which the institute provides. So typically the students, the faculty are very generous in sharing the understanding. So the student is having any difficulty, either using an instrument or understanding the principle of how to use it effectively. So the student goes and asks the concerned faculty, and they are ready to share their knowledge and expertise. So this is essentially how we have been working, and it has worked out well. And then with this, in the examination, during the examination, formal examinations, the professors with other different type of specializations are very open in their comments and criticisms, and these criticisms do help us in understanding the problem. In fact, the way we got into the blood plasma separation problem itself was because there were a sensor group in our institute who was developing these sensors. And then they did not know, I mean, as I had mentioned, that you cannot just take entire blood, you have to separate out and look at only the plasma part. So this is something that there was beyond that domain, and they sort of came to me and said, can you help us with this problem? So that is how the interdisciplinary sort of activities happen in our institute. And there are several other faculty as well who are doing a lot of interdisciplinary work. And there are several centers that I'm going to talk about in the second part of this.
Thank you, Professor. It's very good. It's inspire us. And then the second question is from Yulia Markibra. Dear Professor Amit Agrawal, what about the ideal thickness blood value that property for this system? Because you said that the dilutes for 20 times are good.
Yeah, so initially I showed you for the first device I showed you was 20 times diluted. So we call that as the first generation device. Honestly, we did not even know whether we'll be able to succeed anything. So once we could actually dilute and get something out, we were very happy. Okay. But then we that gave us confidence to continue with our journey. And then finally, the device that I was showing you, the video which I showed you, was with undiluted blood.
Okay. The third question, it's same with the other question. I will read all of them. First one is from Gulam Asrori Buntoro, and then Ruli Retno Muwani. How to manage time in research? The first question. The second one, how long will it take you to research blood cell, and how do you manage your time to research on multivariable of microdevice? I think it's similar question.
Yes. Okay. So again, a great question. So honestly, it took us 5 years to make the device. Okay. So it took us a lot of time. So although the solution looks very straightforward, and maybe we look back at it and think we were fools not to sort of think about it, but it does take that time because you have to understand these things and do a lot of, and I mean, I think that's one of the reasons why other groups have not succeeded, because they have not put in that time and do it systematically. We did a lot of experiments, a lot of failures, etc. And there are several papers, if you have seen, I mean, if you're interested, you can see from my group, where we describe not just our successful experiments, but also our unsuccessful experiments. And we try to explain why those have been unsuccessful, what could be changed. And then slowly we kept on learning from there.
Okay. Maybe more than 20 years, I think. The other question from Sutrisno. As I know from the presentation that the research gap was found from the existing microdevice and the research question was developed as how to make this more simple. What is your advice to find the research gap and research question in the area research of technology management related to this research idea about the research gap, how to find the research gap.
So how do you find the research gap? Is that the, yeah, so that is comes with a lot of critical reading of the literature. I think there is no substitute to that. Of course, there is talking to people who are in that area, and definitely helps. I mean, starting from your colleagues who could be like potential sort of, and a lot of times what I have seen from my personal experience, I can be very honest, what we start off with initially, thinking that's a great problem, we land up sort of solving a very different problem. So that is something that happens because what you sort of realize that once you start off with a research question, you find that either that question is very trivial or very difficult. It's very difficult, very, I mean, only once in a while you find that's a very optimal question that you pose to begin with. So most of the time, either you will think it's a very trivial question and therefore you have to think of something else if you have to sort of think of something reasonable in that area, or you have to think of that, oh, this is too difficult a problem, it cannot be solved. So, I mean, at least I cannot solve all of that in right now, so I will have to wait for some more time or put in more effort to try to solve the other aspects of that. So that is typically what happens. So what we are saying is that you start off with one problem, but then you refine your problem as you go along. I think that's a natural process, and I think that should happen. Yeah. It's very unlikely, although I mean, just to give you an example, just to sort of make, so every student I mean, when a student joins in our institute for a PhD, the person is supposed to give a title of his PhD thesis. And then when you give the final thesis again, then the title is given. And then we look at what we started with the first title and what's the final title, and we always laugh at how different they are. So most of the time we invariably have, I mean, of course it is within the broader theme of whatever it started off with, but but what have you landed up doing is very different from the initial thinking and the plan.
Yeah, I agree with you. It's very not trivial question, I think. We need to dig about the research gap. And then another question from Sidik. From the research about thickness blood especially, why it doesn't use laser with right length of wave provided and photodiode, yeah, as detector of light intensity?
Yeah, so again, as I said, that there are two aspects. One part is the sample preparation and the other one is your sensor module.
Yeah.
So here we are only looking at the first part, the sample preparation module. We are not really looking at the second part. So if you were to look at, so what you are asking for, if I have understood you correctly, is the second part, why use the laser or not use laser. So there are several detection techniques. It could be electrical based, optical based, maybe. So again, so that those aspects we are not getting into right now. That's not really my expertise right now.
Yeah. Okay, bro. And then the last question from Ike Fibriani. Dear Professor Amit Agrawal, may be explained in more detail thickness blood from experimental result using conical microscopy about intensity profile for sample and shield flow rate comparison between experimental and simulation. Thanks a lot, Prof.
Okay, so that's a great question. So, so basically what we do in confocal microscopy is that you essentially image layer by layer. So if you have a channel like this, you image here, image here, image here, image here, and it is supposed to be doing it very fast, very rapidly. So, and then once you get the information, so you then piece up all this information together. So all these different depths that you have, you piece all that information together, and that's how you construct a bigger of the 3D image from the 2D images that you have taken. So that's how essentially the confocal microscopy works. And what you saw was a cross-sectional view where again at different cross-sections, the images were taken, and then you just stitch all these things together. So it comes with, I mean, it does all these things automatically, the imaging at various planes. And then there is a software which stitches all these things together. So all this is part of the package or the part of the instrument that you have. So, I mean, there is nothing that you have to do on your own. It essentially does it for you. and gives you this output. But the good thing is that we do have this confocal microscopy, which is actually a very expensive piece of equipment in our institute, and in fact, a few of them. So,
yeah, okay. That's all, I think. I don't know, maybe from the audience, maybe there is, maybe I have only 5 minutes here. Yeah, about 5 or 10 minutes, maybe there is another question. Instantly, you can write down or raise your hand. Atau ada yang mau pakai bahasa Indonesia? Okay. I think this is the last question. I think we can go on to the another topic, Professor.
okay.
Yes, you may pursue to the another topic.
Okay, thank you.
Yeah, welcome.
So I hope you can see my presentation here. Okay, are my slides visible?
Yes.
Okay, so I will basically be talking about the institute now. So we have already gotten a sense of what sort of activities goes on in our institute. So this is so targeted to any student who may be interested in higher studies, either as a full-time or through an exchange, whatever way is possible. We'll be happy to sort of discuss this possibility, and we'll be happy to explain anything beyond this in the question answer session. So of course, this could have been my very first slide, but I mean, this is happening now. So this is in the map of India. If you sort of, we are just trying to place our institute and our city and then the institute. So we are essentially located on the west coast of the country. And the city is also known as the financial capital of India. So Mumbai, as you may know, is one of the largest cities in the country. The city has a lot of industries, especially the entertainment industry is one of the industries which is very thriving. And a lot of people come to the city with a lot of dreams for jobs and doing well in life. So this is also known as the city of dreams. And we are essentially located in the center of the city. And if you walk in, so this is actually, the city is a concrete jungle. There are a lot of buildings, a lot of people living in the city, but we are actually quite fortunate to have a very green campus. So you can see from some views which are there of our campus. So we have a lot of buildings, but we do have a lot of greenery, a lot of gardens around these buildings as well. We also have like enough sports and other facilities that I will be talking about in one of my later slides as well. So we would say that this is one of the premier institutes which is located in the financial capital of India, and there are 2 lakes and a small hill which are there on the 2 sides of the campus, and one small hill. And you can go trekking up that hill inside our campus. And this is a very safe campus, and I mean, of course, we have a mostly residential campus, which means that most of the students, faculty, staff essentially stays on campus. So that means that in the students community, there is a lot of close interaction that happens. And the students also get to know quite a bit. They have long-term friendship, which is what they develop here because they stay on hostels. And of course, through these interactions, they also learn a lot of other things from each other. So it's a very vibrant sort of a campus that we have with all these activities going on. And the institute was founded around 60 years back with the objective of imparting high quality technical education to the youth. And we have recently been given the tag of the Institute of Eminence, one of the first institutes in the country to be given this tag by the Ministry of Education of India. And the institute has it started off as an engineering institute. So we have all the major engineering disciplines represented in our institute. We have the sciences. We have already seen interdisciplinary work with bioschool. Similarly, we have humanities, design and management as school centers. We also have several interdisciplinary programs, for example, climate studies, Center for Urban Use, machine learning, machine intelligence and data sciences, and so on. So these are all interdisciplinary centers for us where either things have been set up because people from different specializations like interdisciplinary work want to come together and do something, or the center is catering to larger national goal. For example, the Center of Propulsion essentially it supports the defense organization. Similarly, we have the Biomedical Engineering Center. We have already sort of seen examples of that. We have the Desai Sethi School of Entrepreneurship. So this is something which has been named because of the funding which was provided by alumnus. And this is a school of entrepreneurship. So basically students are encouraged to come up with their ideas and then see how these ideas can be translated into products. And what is the journey of an entrepreneur? That's what the center tries to take you through. So in terms of our education, so we can see that in India, at least, we have several million of these students, maybe 1.5 million or so students competing for the undergraduate seats, which are there in 23 IITs. Few thousands of these seats are available. So, and there are a huge number of students who are competing for these seats. And what is seen is that the top 100 rankers, so these students are ranked, the selected students are ranked, and the top 60 of the 100J rankers prefer to join our institute, IIT Bombay, which shows the popularity in the country of the institute. And this happens because we give them a very flexible program with a lot of options of minors, interdisciplinary dual degree program, and so on. We also let the students participate in international competitions, and there are several technical as well as cultural clubs which are there on campus. So the students can be a part of this, and this is another aspect that the students learn and they like. So, moving forward to master's degrees. So each of these departments and centers that I have introduced earlier, essentially gives a master's degree in their field of specialization. And these are typically known as Masters of Technology or Masters of Science or whatever else is the most appropriate master's degree. And then we have the PhD program, which is research-based. We have already seen examples of that, several examples of that, where students essentially does either development work or fundamental research or a combination of both. And it takes typically 4 to 5 years for a student to graduate with a PhD. The first year is typically a coursework, and there may be qualifying exam also. And then, each of these departments and centers would have a PhD program. So moving on from teaching to research. So again, these are some of the facilities that we have. And this is a center I was talking about. We go to the center where a lot of these high-tech instruments are available. Some of these are given here. This is only one center, and we have several centers like this. And even each professor's lab has a lot of these equipment. So we can see that overall the institute, there are very large number of very sophisticated and state-of-the-art equipments. Each lab is very well equipped. So for example, you saw in my lab with the example of PIV, particle image velocimetry, and we have 2 of these, and we can do all these measurements. So we have all these various labs which are very well equipped, and we can do these various measurements, whatever type of measurements that you see that can be done. So I'm just showing the example from central. This is a central facility that we have. So any student, any person can actually go here. So this is not like a individual professor's lab. Now, this was one example that I would like to project about bioengineering. Again, where we have seen these people with their dental implants, for example, with the plasma suppression already seen, and then these devices, I mean, again, making whether it's a flexible robotic hand and such things which people are trying to make, or people have been making and developing and sort of demonstrating it at various stages, technology readiness levels, these are available. So just to give you some examples, some concrete example, this is another professor from our institute who has developed, along with the students, have developed this for audio technology, some, I mean, from listening of music and the speech analysis she can do. So again, there are some sophisticated sort of processing of the signals that can be done, which is what she does. And then these are a glimpse of some of the technologies which have been actually commercialized from our from the various professors' lab. So, for example, this is a telecom router for networking. It's a networking, very fast networking switch, which is employed in, I mean, all these data communications and so on. So this is something which was developed and licensed, developed by Professor and his students, and then licensed to several companies. Similarly, whether it is the COVID time or it is on the other pre-COVID times, you can see these products coming out. So, and the students are also encouraged to, if they want, they can start their own companies. Once you're an alumni or you're connected to the institute, you get a lot of benefits, you get a lot of connections, you get the people, I mean, knowing you outside, and they typically have a very good image about the institute. So you have head start that way. And that has helped form a lot of companies using the technology which has been developed in the institute. And a very large percentage of them have actually gone out to do very well, as you can see from the numbers here. So let me just, so this was to give you an idea of the institute and the type of research that happens. Now I will very quickly show you the campus and the life on campus. So this is actually a movie which has been taken from a drone. So you can, this is really the bird's eye view of the campus. So this is our main building, as you can see. And this is our main building. And this is the Gymkhana. So of course, we'll see a lot more grounds here, the basketball court, the race tracks. This is where the in-house, the gyms, etc. are. Again, a view of the main building. You can see the lake from one of the sides. So this is the cultural night which was taken. And then the sports facilities you can see here. This is the lake, view of the lake. So this is just to give you a sense of, and this is the hill that I was talking about. So this is a hostel that where the students live. So I will stop here. And so it gives you some sense of how things look inside the campus. Some pictures from the cultural events that happens regularly, of course, in the pre-COVID time. I'm talking about the pre-COVID time here. And the sports facilities, we also have Olympic-sized swimming pool, for example. And as I had mentioned, that the students are encouraged to participate in whether it's in the racing, make their vehicles and participate in racing as part of racing teams, or make solar houses, make underwater robots. So students essentially make all these and participate in various competitions. Similarly, there is a satellite, a student satellite which was made, and this was actually launched by Indian Space Research Organization into space. So So that was something which was very thrilling, not just for the student, but for the entire institute community. And this was the first student satellite which was launched from India, from anywhere. So there are several interesting things which are happening on campus. And as we said, that as a student, you get exposure to all these various activities. And then here are some alumni who have actually gone out to do well. So in fact, I would say that anybody who has walked through the corridor have actually gone out to do well, but these are some examples from the non-academic field, because just to sort of show that not just in academics, but in several other areas, whether they have chosen to get into business, or whether to get into politics, or whether to get into filmmaking, okay, they have actually gone out to do very well. And it's only because of the the holistic education that is provided, and the very large, good amount of interaction, contacts, etc., that you make with your peers. All these things actually help you to do very well in your life later on. So moving on, let me just get you some specific things. So if you are a master's students, how do I get admission in there? Essentially, we need a bachelor's degree, which is what would be the requirement anywhere else. And most of the departments also allow students from other specializations into the MTech program. So it's not that if you have done mechanical engineering, BTech, only you can get MTech in mechanical engineering. You can come from other streams, for example, as well. And similarly, so there is a possibility, of course, this is very specific to the department. You will have to go to the department and look up what are the specific requirements. So, but people maybe from maths can come into engineering and so on. So there is a lot of possibility of people from other specializations coming into an MTech program. And MTech programs are typically well-structured 2-year program, which have 1 year of coursework and 1 year of master's project. And out of the 2 semesters, 1 year in the first year that you have, there is set of core courses that one does, and there is large number of electives that one does. And again, for a given elective, you have multiple choices, so that actually you can choose a subject of your interest. The language of instruction remains English, and we start our semester every July end actually. So I mean, close to August is when our semester begins. And we have several fellowships. It's not just for international students. These are especially for international students. So Government of India fellowships are there. So that one can actually avail if you would like. And we can actually share the website details and other things. We can share the brochure where you can get more. I mean, of course, you can, if you remember these names, Steady in India, for example, or ICCR, you can go to their website and look up the details, and we can also share these details through through a brochure. Now, if you were to talk about if you're interested in PhD, we also allow, I mean, typically require master's or equivalent degree to get into a PhD program, but there are several departments which allows even a bachelor's student to get registered directly into a PhD program. For example, mechanical engineering does that. In fact, a lot of my students in the past have just a bachelor's and they have started with a PhD. It again requires some amount of credit, some amount of courses in the first year. And then, of course, we can, there are several examples, as you have seen, for interdisciplinary research. If you are interested, you can have students, supervisors from 2 different departments, and it is possible. So you can talk to a supervisor, and there are a lot of projects which are floating around, and you can register in one of them. So that is also very much possible. And there is a center which is especially does this, the RNTS, which essentially has mandate to bring in people from two different supervisors from at least 2 different departments. So, and there are several scholarships which are available. Study in India, SECR, I've already mentioned. So these 2 are there. In addition, there is a special doctoral fellowship for ASEAN students. So, and this is a very attractive scheme given to from the Government of India again for 1,000 PhDs in all 23 IITs. So this is a very good number that we have. So you can definitely think of availing them. So some of these are still available till the end of this month. So this is, I mean, we had already had several extensions. This is the last extension. So if you're interested, it's still possible. You can still apply under these 2 programs. This one is unfortunately closed now, but the other two are still possible. And the institute also has certain fellowships, so you can directly write to us, although we would prefer that you apply for these two things. So for PhD, we have intake for 2 semesters. We have 2 semesters and we have intake for both these 2 semesters. So whether it's a July, August, September semester beginning in August, or it is the January semester, for both these semesters, we have admissions. So you can apply now, or if you're looking for a PhD, you can also apply later on. Similarly, if you're applying for, if you already have a PhD and if you want to come for a postdoc, okay, that is also possible. We have institute postdoctoral fellowship schemes, which you can go to our website. Again, if you don't find it, you can send us a mail. We will send you the link. And we can also ask us to be considered if you have a PhD with good publication record and all. We'll be happy to even consider you for a faculty position as well. So we have all these possibilities. So which, if you're interested in, we can look at all these options. Now, what we need in terms of documents are very straightforward. In terms, we have just application and any standard material that you would submit anywhere is what we need here. There's nothing very specific that you would see here. So, I mean, if you have applied to other places, you would have all these material already ready with you. And as we said, if you're interested, you can apply before the end of this month, and we can be considered in the next round of admission itself. So some website is given here, and the deadline is the end of this month. And finally, I would say that the institute has been well ranked and well respected, very well respected, although the ranking may not, I would say, adequately reflect that, but our institute, I would very firmly believe, is far better than what you would see here in terms of ranking. We have been consistently ranked as #1 in the country, and recently we have been in the, and we rank in the first 50 in the world in engineering. If you look at subject wise, overall, we are in the top 200. But as I'm saying that, we definitely should be ranked much better than that. But if the ranking matters, so this is what we have to have. So moving on, I think we will just say that we'll be happy if you would like to consider studying in IIT Bombay. We'll be happy to welcome you. India is an old civilization with a lot of different things to learn, and just going to a different land and culture, it exposes to so many other things, and I think it is an enriching experience in itself. So we would be happy to have you consider as IIT Bombay as one of your options if you are looking for anything higher. And as I said, whatever degree you hold, we have an option of, if you want to continue that to a higher level, we have option for that. So, we will be happy to discuss this further, and anything that you would like to clarify, we will be happy to take that. So thank you. So thank you again, and I'm really thankful to Dr. Julian for your patience with me, actually, I should say.
Thank you, Professor. The explanation of the your university. Waiting for another question from the audience. But before that, I think I have some question, Professor.
Sure, please go ahead.
ITT Bombay is founded in 1958, isn't it?
Yes.
And it's similar with our university. The ITS is founded in 1957.
Okay, that's very close.
Very close.
Yes.
But in different country, in different culture is quite different.
Yes.
And my question is, in India, there are a lot of ITT, isn't it?
Yes.
The ITT Bombay is one of them. Can you explain? Can you describe the difference between them, or the historical background that they combine to the one of ITT?
Yes, so great question.
Yes.
So IITs were established in the post-independence. So there were 5 IITs which were initially put out.
Okay.
Now there are 23. So,
Okay.
so 5 IITs, we started off with 5 IITs, and we started off with the first one in 5 different locations, 5 different. So we were like east, west, north, south, sort of a thing that they wanted to go to and have one of them. So we had like around for maybe 40 or 50 years, we were working with only 5 of these. And then people realized that there is a lot more demand for education, and we should be increasing the number of IITs. So then the 6th and 7th, were established, and then suddenly there was like 7 more, and then 7 more established. So, so there have been in 2009, 7 more were sanctioned, and 2014 or 15, and there 7 or 8 have been sanctioned. So now there was 7 original IITs, and then 7 more got added, and then or 8 more got added, and then another 8 more got added. So that is how 7 plus 8 plus 8, yes. So that is how we have now gone to 23 now. Now these are all independent, so they are not. There is no connection between them. We have a common test, but there is no connection after that. So these are all independent programs. They have our own Senate, our own board of governors. So we have independent. We of course, for the bachelors, we have a common entrance test, and there is a common admission that happens. But at the master's level, PhD level, the admissions are not common. I mean, the student can actually get admission in 2 places. For the undergraduate level, the student can only get admission to one of them. They cannot, he or she cannot get admission to 2 of them. So because it's a common portal, so you can choose this seat here or this seat, mechanical engineering IIT Bombay or electrical engineering IIT Kanpur or whatever. So you can choose any of these, but once you have chosen and you have been allotted a seat, you cannot move away from there. For MTech and PhD, you can have admission in masters at IIT, mechanical engineering IIT Bombay, masters in IIT Kanpur, mechanical engineering, IIT Madras, in aerospace engineering, for example. So you can have like admissions, and then you can choose whatever you want. So for MTech and PhD, that is the situation. And so these institutes are all independent. So, and whatever I have talked about is only about IIT Bombay. I'm not talking about the other facilities and other resources and other things that exist in other institutes.
Okay, thank you, Professor. And I have another question.
Sure, absolutely.
You mentioned that at the undergraduate, you have a flexible curriculum. Is it correct?
Yes.
What is meant by flexible curriculum?
Yeah, so flexible curriculum means that they can actually choose do a lot of courses, and then they can actually tag it. So they can tag and retag a course. So for that, for example, they can take one course as an institute elective or a department elective. And then if they want to change the tag of that say, okay, no, it should not be as an institute elective. I want this course to be tagged as a department elective. So then they can move the tag, change the tag. Yes. So, and they can also have large number of electives. They can also have, so that is one part which is there. The other part is that we have a basic bachelor's program. And then we give, if you do more courses, you can get an honors degree. You can also get a minor degree in another subject.
Okay.
For example, you can go with a bachelor's in mechanical engineering with a minor in electrical engineering if you want. That is possible. And of course, if you want to make honors also along with that. So we have these are the flexibilities that we get. And now we are getting a dual degree program. So that means that you can get masters in one, and you can get a master's in mechanical engineering and bachelor's in mechanical engineering and master's in electrical engineering, for example. So you can also do that. So
it's very
you can
flexible.
do a project. Yes. So that's why you have enough flexibility. Yes.
Yeah. Depend of the student what they capable. So is there any other question? Maybe you can raise your hand if you want to ask something about the ITT Bombay or about the topic before. Maybe you have another chance to ask the professor. I have another question, Professor.
Sure, my pleasure.
It's about the company. You said that the ITT Bombay already produced or something like that, produced the startup and company, the entrepreneurship company, I think. How the university do that? I mean, is there any because we are in Indonesia, especially in the ITS, it's quite difficult to produce the startup, to connect the entrepreneur, and then goes to the bring the student to build, to develop the entrepreneurship, to develop the company. We have already maybe 5 to 10, very little than your university. And I want to know, but what the strategy to boosting the to produce that company, that startup, something like that?
That's a great question. So basically, the students, see, I mean, most of it is driven by the students themselves. So the thing is that those students who have entrepreneurs idea, who want to have a startup, they are looking for things that can be translated into a product. So they go to projects which have this commercial aspect, a commercial potential. So that's the first thing that they do. Now, once, and the other sort of, these are the students who have sort of have a pre-plan to go for a startup. There could be also other sort of cases where the student just works on a project, and then we realize that the student and the faculty realize that there is a commercial potential to what has been done, has been obtained. So then they start a startup, which could be jointly with the professor, with just the student in there, and so on. Even an alumni can come to our institute and they can start. So once you have this connection with IIT Bombay, stays throughout your life. So you get, I mean, you can always come back and you can draw upon the privileges. So now what happens is that once you go to that, the society and say that, okay, I have this idea, mostly it has already been protected through a patent, for example. So patent has already been filed.
I see.
So, and for which again, the institute has a patent cell, which tries to help you file these patents. So, and then once so essentially you're trying to commercialize that patent. So then what happens is that this society, which I'm talking about, Society for Innovation and Entrepreneurship, they look at the idea and they say that they will ask you to do some market survey and say whether it is worth taking it to the market, etc. Who would be the potential buyers, who will be putting the money, and so on. So we essentially ask you to do this journey, and then once they're convinced, then they will let you incubate your company in there. So they will provide you some facilities, connects, etc., etc., real estate, etc. So all these things are provided, or at least some things are provided, and then you start off from there. So I think a lot of screening happens, and because the technology itself is to begin with is supposed to be good, and that's why we see much higher success in our case. In fact, most of the time, if you know that the 90% of the time, more than 90% of the startups fail actually, and in fact, within 90% fold within 3 years is what they say, and 99 or 98% fold within another 5 years or so. So it's only 1 or 2% of companies that survive, whereas what we see in our case is a much larger percentage. So it's just because of the lot of pre-screening and the quality of the technology to begin with, which was very sound, that has helped to for these companies to be far more successful.
Okay. Yeah. Okay. Okay. Thank you, Professor. It's inspiring.
Thank you for your nice question. I mean, yeah.
We have another question from Nigeria, Professor. Masud Kabir, Mr. Masud Kabir, from Audu Bako College of Agriculture, Dambata, Kano, Nigeria. How are students handled with recent COVID-19 situation, Professor? Will a student get visa in this time?
Yeah. So what is happening is that we are essentially asking any student, whether it is our Indian student or a foreign student. So if the student is resource constrained, for example, there are issues with internet connection, power connection, power supply, or anything else. I mean, sometimes we find that the student does not have a steady room in his house,
Oh
for
yeah,
example.
yeah.
So that's a major problem because the houses were not designed like that. So in those cases, the students are writing to us, and there is a separate committee which has been trying to get these students. So in fact, a lot of our foreign students either did not go back or they have come back. So in fact, our foreign students are mostly on campus again because, I mean, if the moment they would write to us, we'll say that, okay, we would be happy to have you back on campus. Of course, you have to comply with all the government regulations. You have to take care of all the norms during travel. And you have to quarantine yourself after coming to the institute. So those are well laid norms and guidelines that you have to follow. But from our side, from the institute side, if you have enough justification for coming back on campus, we are allowing these students to return. And as I'm saying, a very large percentage of our foreign students have actually come back on campus.
The lecture already on campus or just hybrid or just online, still online?
Yes, right now it is online. We had a very deadly second wave, so we are still going past that. So it is it's on the lower side, but it's still not come to a very low level. So it's still quite high actually, the number of cases in the country.
Yeah. Okay. Is there any other question? Bapak, Ibu, monggo. If you have any other question, you can raise your hand. We don't have? Three, two.
Any clarification if you want on the institute, I would be happy to clarify. Yeah.
The time is still. Ms. Nur, how about the time?
Actually, we still have several minutes left, but it's up to you if we want to change the plan.
No, I think there isn't any other question, I think. If you have another question, maybe you can, and Professor, maybe, there's one raised hand. Afifatul.
Okay.
Yeah.
Okay. I'm sorry I cannot open my camera. It's okay. Thank you for your explanation, Prof. I wanna ask about. I have been searched in your. In your website of IIT Bombay. It said that we did we need to have a professor approval before we apply for PhD? And then the another question is, here said that there is TAP and FA topics. What is that? Is that the topic for our dissertation or what is that? And then that is what do we need the approval from the professor from the IIT Bombay for the PhD application?
Okay, so see, most of the departments don't need that. So you come to the department as a student, and once you are registered in the department, you can choose a professor and you can choose the topic of your dissertation. So there is no need to contact the people beforehand. Of course, if you want, you can do that. You can be writing to somebody, and it helps. I mean, it can help. I mean, there is no harm in doing that. But we would always say that, at least I personally tell anybody who wants to who contacts me, that you first come here, you look at all the options, because there are a lot of projects which are being floated. Look at all the options and then make up your mind. Don't just sort of make up your mind because you have heard something good or bad about somebody, okay? Because that impression may be a very narrow view. So you come here, look at the things firsthand, talk to people, talk to the professor who you want to work with, and then make up your mind. That's a much better, more, you are then taking an informed decision rather than taking some second-hand, third-hand information. Don't go with that. And most of, I mean, the good thing is that most of the departments do allow you to do that. So they let you just come in and then after that, pick up a topic and pick up a professor. So I hope I have answered that. And then you had another question about, which I did not. Can you, if you repeat the second question, second part of your question?
The TAP and FA topics in your website.
Okay, so there are several.
The chemical engineering department here.
Yes, so essentially, yeah, so there are two sort of ways. One is that TAP essentially means that this is TA through a project. So that means you join a particular project beforehand. So there is, so for example, if I get a research project and I'm looking for a student, okay, then I will float that as a topic and say that somebody who is, I mean, only that student can come for that. I mean, whoever is interested and whoever I select can only come for that topic. Okay, so this is a TAP seat. So here you are looking at the professor beforehand, talking to them, and the professor thinks that you are a good match. Your expertise, your background is a good match to what is required in the project. And therefore that student will be offered a position in that project. So that is one thing. And the other thing is that it's a general admission that you get. So for foreign students, we will be having all these scholarships that you see. That scholarship is not related to any, it's not tied to any project. So that means once you get an admission, your admission is confirmed. Okay. Once you have your admission confirmed, you can join the institute and then look up for a PhD topic and a professor. So this is much better. I see that this is much better because it's not that, because once you come here, once you come to the institute, look at around, talk to people, you get a much better sense of what you're interested and what you're not interested in. You can look at the professor's lab. You can talk to people who have graduated or who are graduating and understand how the professor will work with you, how much time he gives you, how much detail he spends. And I mean, how much detailing, how many project papers, etc. Whatever you're looking for, you can actually make a inquiry and then make up your mind. You don't have to commit to anything beforehand. So I think that's a lot of advantage that you get. It's in your own advantage that you have that flexibility. So I hope I have answered your question.
Thank you, Prof, for your explanation.
Thank you.
Okay, Is there any other question? How much the living cost of in Bombay, Professor?
How much? Sorry, how much is
the living cost? The living expensive.
Yes. Okay. So yeah, I should have actually mentioned that. So what we give actually as a scholarship is actually is more than sufficient to cover cost for one person. If you're coming with a family, it becomes slightly difficult, especially because accommodation in Mumbai is very, very expensive. And accommodation for married people is very difficult on campus. So that is the constraint. But if you're coming as an individual, we have accommodation. We will get accommodated in a hostel. So, and your cost of living will not be very large, and you will be saving enough. I mean, typically the students save enough I mean, enough means enough. I mean, they're
Yeah.
a very good percentage of their actually a stipend they land up saving. So it's actually a very generous scholarship is what you get, is what I would say.
Very good. Is there any question? Okay. I think that's all, Professor. Um, your material, your subject, your topic is very good. It inspires us, and thank you very much for your sharing. I will give the time to the MC again. Ms. Nur.
Okay, thank you very much, Professor Amit, for the excellent lecture today. And thank you, Mr. Julian, for conducting this amazing session. Please give applause to our speaker and moderator by using the Zoom reaction feature for today's amazing session. Okay, and then we would like to present a certificate for our honorable speaker and moderator. Okay. Okay, so this is the certificate for Professor Amit Agrawal. Thank you very much,
Thank
Professor.
You. Great honor. It's really nice for me to be here, first of all, get this opportunity to talk about my work. And thank you all for your wonderful way of conducting this session. So I should say thanks to Dr. Julian for his great patience.
You're welcome.
You're welcome, Professor. And I also want to thank you for your availability on today's session. Okay. For the next certificate is for Engineer Durendra Bambang Aryateja for being our moderator. Thank you very much, Mr. Julian.
Thank you very much. Thank you very much for all your committee. Thank you, Professor, again. Thank you very much.
Thank you.
Inspire us. Thank you.
Okay, you're welcome. Okay, before we end today's session, we invite you all participants, as well as the honorable speaker and moderator, to take a group photo. Okay, so for the participants, please open your camera. And maybe we will stop sharing now. Okay, I will wait. Okay. Okay, so I see how many slides? 6 slides here. I will count when some of you are ready to take the photo. Okay. Okay, I will start taking the photo now. Maybe the other committee take some too, so we will have another backup. Okay. Okay, for the first slide, 3, 2, 1. Next slide, 3, 2, 1. Keep smiling because maybe you are in the last slide. 3, 2, 1. Okay, 3, 2, 1. Okay, second to the last. Wait. Okay, finally last slide. Okay. Now we have finished the group photo, and then for the participants, please fill the feedback form through the link bit.ly/feedback _R2SEP 2021, and you can also see it on the Zoom chat room. We want to remind you that the participant who will get the stamp for it is the participant who come on time during this event until the end, and also fill the feedback form. Finally, we have reached the end of today's Researcher and Research Student Enrichment Program, and we sincerely apologize for any mistakes that we may have made during presenting as master of ceremony and the committee as well. Thank you very much to our speaker, moderator, and all participants for the attention and cooperation. Before we part, I would like to inform you that we still have several future agendas on R2SEP for hard engineering stream. You can see it on the poster we are showing you right now, or you can check it out on ITS Global Engagement website later. We will see you again in the next session on the 3rd of June 2021 with the theme Getting Published that will be delivered by Dr. Natarianto Indrawan from the Department of Energy, USA, and it's on 7 p.m. GMT +7. Once again, thank you everyone for today, for the speaker and the moderator as well. Good evening, and wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
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 Thank you, Prof.
Thank you. Thank you. Bye.
Thank you,
Terima
Professor.
kasih,
Gamal.
komite.
Thank you, Mr.
Kawan-kawan.
Andi Mehta. Thank you for coming.
Pak Asto ya.
Terima kasih Pak Julendra. Yeah.
Ya, sama-sama. Saya live ya.
Okay, thank you. I will give it back to the host to end the meeting.